This is a presentation that I've given at soft conferences for more than 25 years, and it hopefully changes every year with some updating. The talk I'm giving today is somewhat similar to the one I gave last year, but I've updated it, and I think I've improved some of the slides. And thus, I think that even though last year's is probably still posted, Kelly and Terry, we might want to replace last year's for this year's, because I think my slides are ideally more clear. So my talk is divided into these three parts. Most of the talk will be what I like to refer to as a primer on chromosomes. That is a learning experience about those human structures, those structures in all animals and plants that we call chromosomes. I'll spend a very brief amount of time on an overview on trisomy 18 and 13, because there are other talks about that, but I do want to say a few words to kind of make sure we're all on the same page as I close with just a checklist for routine healthcare supervision and follow-up by specialists and primary care practitioners for individuals, children and adults with trisomy 13 and trisomy 18. So I do like to include especially that last piece for those that haven't heard this talk before, so they can ideally go back to their primary care doctor or nurse practitioner and kind of review what the checklist of routine care is. So here are the basics. We are going to talk about genes and chromosomes, and I hope at the end of this, I'm going to do a little summary vocabulary lesson. I hope that you actually incorporated some of the vocabulary words. I am sure that many of you who are listening have already heard most of these words, some of these words, and many of you have already heard some of this basics, but to put us all on the same page, I'm going to give you a medical student, a graduate genetic counseling student level discussion of chromosomes with some mention of genes. So here is the famous DNA molecule. As you can see, it's the twisted ladder. It's deoxyribonucleic acid. And interestingly, the little bars that you see horizontally that are different colors are actually the base pairs, the four basic base pairs. The names of them, the A, T, G, and C, are at the bottom. I won't go through the actual biochemical names of them, adenine, etc., but they make up the backbone along with the phosphate of this twisted molecule. And as you probably know, a gene, G-E-N-E, a gene is really a finite defined strip of DNA. And we'll talk more about that in a moment. And of course, genes are what are packed in chromosomes, and primarily what we're going to talk about more today are chromosomes, but of course we have to start with genes. So something to know, and again, those of you who have taken probably high school or perhaps maybe a college biology class know that this is the basic premise of biology, whether we're talking about fungi, we're talking about RNA in a virus, or we're talking about a mastodon, or we're talking about a human being. This is the same, and that is that DNA, deoxyribonucleic acid, replicates and is then transcribed, which includes a process of trimming it down into ribonucleic acid, which then forms the blueprint to be translated into all of the proteins in our body. DNA to RNA to proteins. And as you know, proteins are what do everything. So when we discover a gene and everybody gets excited, we found the gene for this form of breast cancer or cystic fibrosis or this particular condition causing autism, we get all excited. But what we really want is for that gene to lead us to the protein. And maybe if we can work somehow on the protein in the cell, maybe we can treat the conditions. So genes, you can see the top panel there, the same picture of the twisted molecule, by the way, only characterized in 1953. That's honored every year with DNA Day in April, when that particular paper was published from the authors who were at the time in England. Anyway, the DNA is twisted together, wrapped together, and packed with a number of protein molecules. You can travel down this path as you go along into a chromosome. So a chromosome is made up of the genes packed together, but then stored in a very specific way with all of these other protein, these spheres you can see there. Really, most of what you see in this diagram was not really known about till the last 10, 15 years, even though DNA was known about in the 50s and earlier even, and chromosomes were really characterized also in the 50s. So here's the primer. So here is a karyogram, that's with a K, karyogram, which is a depiction of all 23 pairs of chromosomes, totaling 46, 23 pairs in the typical person, with down in the far right-hand corner, the sex chromosomes, in this case, 2 X's in a female. This was a male karyogram of a male. It would be an X and a Y there in that lower right-hand corner. And they're arranged in an order from the largest to the smallest, largest being up in the upper left, number 1. Maybe you can see my mouse, down to just about the smallest, the number 22. Actually, the number 21 is the smallest, other than the Y. But we didn't know that back in the 60s when this was characterized, because they couldn't tell the difference between 21 and 22. 21's famous, of course, because when it is 3 copied, that is, when it is trisomic, causes Down syndrome. So here's a karyogram. It's performed by drawing blood on a person, taking the cells from the white blood cells out, stopping them in the middle of their growth phase on a slide, and then breaking it open with a salt solution. So all the chromosomes spill on the slide somewhat randomly. And then it used to be we would cut them out and paste them on a whiteboard, but now it's done by a computer, and they're drawn and put in this specific way. So you can see that they're curled and they're turned over. Some of them are straight because this is how they spilled out randomly in this particular girl or woman's chromosomes when they made them up. Notice on number 15, there's even a something black mark across, because that was actually another chromosome that didn't quite separate from the other chromosomes. And you can see that as well. So you can kind of see the noise, if you will, of that particular process. So all the genes are lined up on the chromosomes from the top of number 1 to its bottom, and on all of the chromosomes, all 22 non-sex chromosomes. The word for non-sex chromosomes is autosome. I'll show you that word in a few moments. And then again, as I said, the sex chromosomes over in the corner. So up until the early 1980s, we didn't know where genes were on the chromosome. We knew that some genes like color blindness or hemophilia, another 100 or so, were X-linked because of their family tree pattern. But we didn't know where they were on the X chromosome, or for that matter, on the Y chromosome. We knew a few, but I won't go through that in detail. But with DNA techniques of the late 70s, early 80s, we mapped many human disease genes to their chromosome. And then by the 90s, we started identifying the genes and discovering them. So now we actually know 4,000 diseases whose genes have been identified. And that's roughly about 1/5 of the predicted 20,000 genes that are thought to occur all dispersed through all of these different chromosomes. So just when I give this talk in person, I usually ask people to maybe ask me about a particular human disorder. Maybe I will be able to tell you where the gene is. Can't always do it. Sometimes I can, but just to give an idea, the Rh blood type is on the end of the number 1. The ABO blood type is on the number 9. The cystic fibrosis gene is is here in the middle of the number 7. And of course, just what we're interested in, here's the 9, especially for trisomy 9 mosaicism or partial 9. Here is the 13. And of course, here is the 18. And as I mentioned, 21. Notice the stripes on the chromosomes. They're called bands. Every chromosome has its own banding pattern because the stains that they use, and there are several stains, I won't go through them, the stains they use to sort of arrange this for us are non-randomly connected with the DNA that's on that chromosome. And so that's why the bands kind of stain differently depending on the stain or depending on how they're lined up. But if we just take this particular white band here in the lower part of the 1, that's not like one gene, of course. That's probably hundreds of genes, but they're all within that particular stripe that I pointed out on 1. So here's a cartoon from Jordy's textbook of medical genetics that's showing you kind of how they're generally shaped. The number 2, the middle part of the chromosome called the centromere, which is here. I didn't put that on my vocabulary list, but the centromere kind of in the middle right here. The centromere divides the chromosome into its shorter arm called the P, from the French petite, and the longer arm, the Q. So this is the P's and Q's of chromosome anatomy, if you will. So notice that some of them, like 1, 2, and 3, have their centromere in the middle. So the short arm and the long arm, pretty similar in size. While 18, chromosome we want to know something about, the centromere is kind of placed upward near what they designate as the top part. And then here in the 21, these short arms here don't actually have any active genes on them. They're actually called satellites, and this is called an acrocentric chromosome. 13 is an acrocentric chromosome, and we'll talk more about that in a moment. So here from the cartoon, you can kind of get a better idea of these stripes because it kind of brings it out for you. But depending on where in the process of cell division you stop the division to display your chromosomes, you get longer and shorter chromosomes, and you also get more or less bands, and you'll see that as we go through examples. So here's what we care most about, what chromosomes do. And so people who work in chromosome biology and medicine, cytogeneticists, divide chromosome conditions into various groups, conditions that are due to changes or alterations in number, a full change in number, a trisomy. That is an example, trisomy 18, or if there's an extra chromosome, or if there's a full missing chromosome, monosomy. So those are conditions in number: Down syndrome, trisomy 18, trisomy 13. Turner syndrome is an example of having only one X chromosome. Then there are conditions of structure, which are related to partial trisomies that you hear about in related disorders. 9p trisomy being an example of a condition that we're interested in. And so these involve parts of a chromosome that are missing, deletions, or extra duplications, synonymous with partial trisomy. And then there are rings, inversions. I'm not going to talk about them. And importantly, translocations, which can lead to missing pieces or extra pieces, partial trisomy, partial monosomy. And we'll talk about that in a little bit more detail because it's an important cause of partial trisomy 13, partial trisomy 18, and of course, 9p trisomy. In addition to conditions of number and structure, another way that you can look at this are conditions of autosomes, that is chromosomes 1 through 22, including 9, 13, and 18, and conditions of sex chromosomes, of which I just mentioned. One of them, Turner syndrome, which involves having a single X rather than having two X's. But we're not going to talk about the second type of classification today. So there's a karyogram up on your left, again, this time of a female. In this case, showing, if you see, look down, there are 3 18s in this particular female. And here's a nice cartoon of the 18 displaying the bands for us, and also displaying the way these bands are numbered. Here on the P arm, the P, or on the Q arm, the Q, and they're numbered by segments and then by a particular band. So that this particular designation here of 18P11.31 means that it's in the first segment. There's only actually one segment of the short arm of 18, and then it's in a subband that's called 0.31. So there've been conventions that occurred in the 1970s, 80s, and 90s that named all the bands. And then as I got better at defining them, then they changed even some of the naming system. So there are probably 400 plus genes on the number 18. And if you have any desire to actually know all of the known 400 genes on the 18 chromosome, you can go to the OMIM database. That's a free access database. If you Google OMIM, you get in, and there's something they call the morbid gene map. You go in, you pick your chromosome, and you can actually go from tip of the 18P to the 18Q and see every gene. And then there's a link to what that gene is known to do, or sometimes known to cause a particular condition. So here's again our cartoon of 18. Here you can see the bands again, P11.23 being right there as being designated, but here's 11.32 is the white band there, 0.31 is this band, 11.1 is this band, and then the same in the long arm. So it turns out that people have been studying both in trisomy 18, trisomy 13, partial 9p trisomy. They've been studying what part of the segment of the chromosome, short arm, long arm, and thus the genes, are involved in causing the syndrome. That is, what do you need to have to be extra? What genes to have the Edwards syndrome or the Patel syndrome of trisomy 13? So it turns out in children who have partial trisomy 18, over the last few decades, have allowed the investigators to kind of get a feeling when you compare that child's pattern to classical Edwards syndrome, you can get a feeling for what part of the 18 has to be duplicated, has to be trisomic for you to get Edwards syndrome. So most of the Edwards syndrome is seen if you have trisomy for 12.1, this dark band right here. That's about, makes up more than 3/4, makes up 80% of the long arm. Turns out that if you have partial trisomy of the 18 short arm, you don't have hardly any component of trisomy 18. The children have very non-specific facial features. They have normal lifespans. They have mild growth problems, and no birth defects. So it seems like that most of what's in Edwards syndrome is down here in the long arm. But interestingly, if you look at children who have this whole long arm without the short arm, they don't quite have typical Edwards syndrome. They tend not to have the same risk for serious medical problems, although they do have similar developmental disabilities. Anyway, another conversation, but interesting part of the story. But point is, you need trisomy of probably all of the 18 to have the full Edwards phenotype. But if you have most of the long arm and part of the short arm, you probably have almost all of Edwards syndrome. So what is the reason for trisomy, whether it be 21, 18, 13, or for that matter, even trisomy 9, usually trisomy mosaicism? Well, the reason is not known exactly why, but the process is called non-disjunction. That's on our vocabulary list, non-disjunction. So here's a nice diagram of this, which is meant to show you. So if we're going to travel down this path, the one on the left first, and here you can have a person that's going to make egg or sperm. Doesn't matter which parent it is. We'll just say the father for a moment, and you can see two different colored chromosomes. One of these came from this parent's grandmother, and one came from grandfather. Well, the first step in making sperm and egg, and that step process is called meiosis. And there are two steps, meiosis 1, meiosis 2. The first step involves breaking apart the two chromosome pairs. So that one goes one way, one goes the other. But notice here on this panel on the left, they both went to the same one. And this cell didn't get any 18. By the way, all the rest of the chromosomes are theoretically there, but we didn't diagram them. In meiosis 2, the chromosomes split from top to bottom, longitudinally, down from the tippy top to the bottom, and then you end up with this, what they call chromatids now, here and here, and here and here. And you can see these two gametes, future sperm, because we're talking about a father, these two gametes have two 18s, since we'll just use 18 as our example. And of course, if both of these are then joined with a partner's gamete that only has one 18, that particular offspring has three 18s. So that's the process of non-disjunction. It either can occur in the first step in meiosis, or it can occur in the second step. I won't go through the second step in detail. Probably most of you learned about meiosis, maybe even in high school. There's even other little sub-steps involved, which I didn't go through in detail, but it is interesting, and it's why our sperm and egg have half the number of chromosomes than all the rest of the cells in our body, and why most of us end up with 46 or 23 pairs of chromosomes. Now, why non-disjunction occurs is still not known, and there are thousands of PhDs who have gotten their degrees over the last 5 decades trying to study this process, which was originally observed in fruit flies. I mean, they actually observed it in Drosophila. We assume it in humans we just see the end results of changes in humans. We don't actually see non-disjunction. Of course, we know that parental age is a factor, maternal age more than paternal age. But otherwise, we really don't know very many factors involved in non-disjunction. There are no environmental agents that we know about. People have postulated various, but nothing has ever been actually proven to be the case. Some people think that one of the hypotheses is when the chromosomes line up, they exchange material. In this first part up here in parent, they have to exchange material. That's called recombination or exchange. And in that process, maybe something happens that makes it where they don't separate like they're supposed to. And there are some human conditions that involve lack of separation, like non-disjunction occurring in a different way. But we still don't really understand non-disjunction. It really is, in that sense, kind of a random event. But it is the basis for why trisomy 18, trisomy 13, and really even trisomy 9 mosaicism occur. So chromosome 13, this is an acrocentric chromosome, meaning that the centromere is toward the top, and this satellite here doesn't really have any genes in it. And that becomes important in the common chromosome 13 translocations that we're going to talk about in a moment. So notice the cartoon again, but notice here, the cartoon on the right has more bands because they stopped it in the separation process earlier in the process. And thus, when you do that, they time it in a very specific way, you actually get more bands. So it allows you to define the chromosome better. And this technique of being able to detect smaller and smaller missing pieces has really primarily occurred just in the last 20 years. And then now, you may have heard this, they use this technique for chromosomes called cytogenomic microarray, or shortened to microarray, which really has to do with better ways to define how much DNA is missing or extra. Anyway, back to 13, acrocentric chromosome. Again, people have been studying different segments of the chromosome that allows to having a full Patau syndrome or a partial trisomy 13. So if someone has trisomy for the top half, they do not usually get an orofacial cleft. The person also usually does not get extra digits of the hands and feet. Well, if there's trisomy for the bottom half of the 13, the child has a lot of what we know in the Patau syndrome, that is the extra digits of the pinky side of the hands and foot, and also the orofacial cleft. So children with partial trisomy 13 of the bottom half resemble and overlap a classical Patau syndrome more than those who have trisomy of the top half. Again, that could be a whole other talk, but important to note. So here's a karyotype of a boy. Notice there's the Y chromosome. A boy, down right-hand corner, with trisomy 13, having in this case a full complete extra 13. And it turns out that in trisomy 13, a little more than 90% of children and adults we know with trisomy 13 have full complete trisomy 13 with non-mosaicism, every cell with 47 plus 13. A little less than 10% have either a translocation with partial trisomy 13, always of the long arm, or mosaicism. So mosaicism makes up about half of that 10% that is not full complete 13. In trisomy 18, full complete trisomy 18 is higher. It's more than 95%, and only about a few percent of people, 2%, who have a lot of the Edwards syndrome or most of the Edwards syndrome physical have partial trisomy 18. A lot of other children we know with partial trisomy 18 don't even have very much of the Edwards syndrome because they have a smaller extra piece. But back to 13. So translocation becomes an important discussion in trisomy 13, also 18, but numerically not quite as much. And the most common translocation in trisomy 13 involves the most common translocation in human beings, a 13-14 translocation. And you can see the middle picture here where the 13 and 14 are on the left-hand side. That's the different stripe patterns, 13 and 14. They break apart at that centromere, and then they come together from long arm to long arm and form this fusion 13-14. Well, 1 in 1,000 people have a 13-14 translocation. So you think about a sporting event, when we used to be able to go to sporting events in person, now we're getting back to it, but think of a sporting event, 50,000 people in the stands. Most of them don't, many of them in the stands, maybe as many as 1%, carry a 13-14 translocation, and they don't know it. And they don't know it because most of the time the risk to actually have a child with trisomy 13 is only a few percent. So it turns out that they never find out that they have the 13-14. I have a number of examples of medical students who did their chromosomes as a sophomore medical student or even as a pediatric resident and found out they had a 13-14 translocation. A friend who didn't know that she had a 13-14 except for a serendipitous reason. So this is important to know about because the 13-14 is the most common translocation of partial trisomy 13, but of course the partial is really almost full. It's called Robertsonian because it was described by someone named Robertson. Notice I forgot to edit from this original slide in French. That's Robertsonian or the derivative, that something we'll talk about in a moment. So here you can see is an example of a person, in this case a female, XX, who's carrying a 1314 Robertsonian translocation and thus is at risk for that passing on with, in a certain way, for the child to have a 1314 translocation type of trisomy 13. There are also Robertsonian translocations of 15 and also of 21 and 22, but we're not going to talk about them today. We'll use this as our primary example. But notice, I'm sure you didn't have time to count it up, but if you count up the chromosomes, there's only 45. There's not 46, because the 14 over here is on top of this 13 in this fusion chromosome. So this person has two 14s, that is all of the long arm, and two 13s, but they only have 45 chromosomes because of that fusion chromosome. Turns out that their offspring, if he or she were to have this type of translocation trisomy 13, they would have 46 normal chromosomes because they'll have this fusion chromosome with the extra 13 attached. And that's shown here. Also a picture from Dr. Jordy's textbook. This is actually quite nicely done picture of bands. You can see maybe the most crisp bands we've had on any figure today. You can see how easy they are to see compared to some of the other pictures. But here, this particular boy, you can see has one 14, two regular looking 13s, and then the fusion chromosome of the 13 with the 14 on top. So that this particular child has, if my mouse is visible, 3 13s because of the two freestanding, and then the third with the 14 on top. And so I think in the interest of time, I'm not going to go through this picture with you in detail because it takes a while to go through it. Interestingly, I did this the last few days. You can Google this and you can find these diagrams, but this diagram is meant to show you that when someone carries a 1314, the possibilities are that they can have someone with a normal 1314, typical if you will, someone with the fusion chromosome would be a carrier, or someone who has trisomy 14, monosomy 14, trisomy 13, or monosomy 13. But of note, it turns out that only 1% of the time does that carrier person have an unbalanced 13. And of course, 14, monosomy or trisomy, usually results in a pregnancy loss. But the 13, the one that's actually here, only occurs in about 1%. So that's why so many people being a carrier, but actually not know about it. So that was kind of the the A's and B's or the P's and Q's of Robertsonian translocation leading up to that important cause of trisomy 13. It doesn't characterize all of the causes of trisomy 13, but certainly the most common. The other translocation, besides the Robertsonian translocation, is the reciprocal translocation, which is the one that would result in most individuals that would be called partial trisomy 18 or partial trisomy 9, or even in some cases that are not due to the Robertsonian translocation, partial trisomy 13. And this is a beautifully diagrammed picture of a reciprocal translocation where the two chromosomes on the left, and this will be a 3 and an 18 for purposes of discussion, where a very significant piece, about half of the number 3, moved over onto the 18 in the top part of the long arm, and that part of the 18 where the other piece joined goes over to the 3, and now you have this reciprocal translocation where you have an 18 over here on the 2 on the right here. You can't see my mouse, 18 here. And there you have the 18 that's got this portion of the 3 the bottom. So this is the basic idea of a translocation leading to a partial trisomy 18 or partial 9p trisomy, if we were talking about that. And in all of those situations, when you get what they call the derivative chromosome, the DER chromosome, that's the word for the derivative, that is the rearranged derived chromosome. The person almost always has both a partial trisomy and a partial, could be small, missing piece or deletion. And again, we're not going to have time in what we have to cover, spend the whole lecture just on what I've spoken about, whole seminar in the last 10 minutes. But in the interest of time, we'll leave it at that particular point. But it hopefully provides the foundation for an understanding or comprehension of translocations both in 18, 13, 9, and other chromosomes. And here's an example of a 9 and 22. Again, I won't go through it in detail, but it's meant to show you the same idea. So here's the vocabulary I want you to think about, or at least hope that you might have taken away from. I could have added karyogram, which is the picture of the chromosomes, or picture of the blood test called a karyotype. But these are the terms hopefully I use that you might be able to walk away with. When I told you about the meiosis and the different stages of meiosis in non-disjunction, I forgot to mention that what's interesting about meiosis in trisomy 18 compared to trisomy 21 and trisomy 13 is that most of the time the change or error, if you will, in meiosis in Down syndrome trisomy 21 and in trisomy 13, 99% of the time is in meiosis 1. But for some reason in trisomy 18, and they can figure this out by DNA labeling, in trisomy 18, about half the time it's meiosis 1 and half the time it's meiosis 2. That must be telling us something, but honestly, we've known that for 2 decades, and we still don't know what that particular finding means. Anyway, some of the other, here's the word Robertsonian again, reciprocal, and mosaicism. So I'm positive all of you are quite familiar with the term mosaicism. It's really where there is the presence in a person and the tissue of 2 different cell lines, could be more than 2, but usually 2, that are derived from a single zygote. And zygote is the fertilized egg. That is the combination of the sperm and the egg. The moment the sperm and egg are fused, that time before any division occurs, that's called the zygote, the fertilized egg, if you will. So when that zygote goes on to form an individual, if there are more than 1 cell line, there are different ways that can happen. That's how mosaicism, that is the definition of mosaicism. Most of the mosaicism we're interested in is called somatic, which means that it occurs in the body. That's all the children and people we know with trisomy 18 mosaicism or trisomy 9 mosaicism. But what's also important is that sometimes there's mosaicism, like more than 1 cell line, just in the sperm and egg progenitors, just in the testes or the ovaries. They call that germline mosaicism, and that's thought to be the basis for the rare time that we see a family who's had a child with a trisomy have a second child with the same trisomy. It's thought to be that there might have been germline mosaicism. So I'm going to stop at that point because I wanted to actually be done with my primer just at this point. And I do want to leave a minimum of 10 minutes for questions. So what I'm going to do is I'm going to recommend, refer everyone to when the talk is posted online, which will be a few days or early next week when it's posted online and replaces last year's talk, which is similar, to make sure that you look at the last 10 minutes of the talk in more detail, because I don't want to simplify the next part, because I really want you to walk away with one message of this part, but I do want to leave at least 10 minutes for questions. This young lady is more than my placeholder here in between the talk. She's this time she's late 20s, trisomy 18, engaging, smiling young lady with her sister there on the right. They're from Sweden, and some of you, if there are any people here that still like to attend this talk over the years, this family came to SOF for a number of years, but haven't been in recent years. And she passed away when she was in her early 30s. So, like I said, I'm going to do this rapidly and refer you to the talk and give you this brief overview, because I do want to at least go through the checklist of healthcare supervision, because I want the people who are listening to this for the first time to at least have that checklist for their doctors or their nurse practitioners. So trisomy 18 occurs in about 1 in 6,000 liveborns, 1 in 5 to 8,000 liveborn babies. It's a recognizable pattern of birth defects. And there is Sarah, the first patient described by Dr. Edwards, and that's why it's called Edwards syndrome. She was in his paper in 1960, and he told me her name, and that's why I know that. And that's Dr. Edwards on the right, with his wife on his right, and that's Kyle down there, who's now in his early 30s with trisomy 18 mosaicism. This was at the picnic at St. Louis SOF meeting in 1990. And Dr. Edwards passed away a few years ago. Again, I'm not going to go through the trisomy 18 syndrome. Probably most of you are familiar with it. And there's Carey Holiday up there in the top panel with Rachel, first two girls whose mothers formed SOF in 1979. And one of my little friends from SOF in the lower picture, and her picture is in the textbook by Jordy called Medical Genetics. There's Chris Holiday. For those that haven't gotten a hug from her, you will at some point in the future when we have a live SOF meeting. Trisomy 13, third most common autosomal trisomy, about 1 in 12,000 newborns, also a known pattern. You know, as you know, clefts of the lip and palate, small eyes, the extra digits are polydactyly. And there's Dr. Patel's first patient, reported in 1960. The paper on Dr. Patel's patient was in the same issue of the medical journal as Dr. Edwards' paper in 1960. There's Aaron, one of my longtime friends with trisomy 13, who I knew over the years coming to SOF meetings. He's actually, his family were kind enough to have his picture in Jordy's textbook over the years. Skip that slide. I'm going to skip the next two slides about number of case reports of older kids with trisomy 18 and 13, and get to what I wanted to be the last two slides, which is just meant to, there are places on trisomy.org where this is available. There are numerous medical articles, including the article that I helped write in 2012 that's also on trisomy.org, but it's meant to be sort of like the checklist of how primary care doctors, family practice doctors, pediatricians, nurse practitioners should be initially evaluating and then following children with trisomy 18. Of course, cardiac evaluations, it's 80 to 90% of children with trisomy 18 and 13 have anomalies of the heart. An airway assessment, which is important to be early and comprehensive. And Dr. Glenn Green will give the professional talk on Thursday. And since I know parents now can attend that, he has given 2 SOF talks in the